A tumor is the physical outcome of a systemic breakdown in the molecular machinery that regulates cell survival. While a growing mass appears chaotic, its formation follows a specific biological logic, the step-by-step sabotage of cellular safety protocols. Our genome contains highly conserved, redundant self-destruct mechanisms. These are evolved defenses, specifically tuned to identify and eliminate rogue cells. This raises a central question of oncology. How does a cell bypass billions of years of genomic failsafes to achieve immortality? Understanding and eventually stopping this process requires us to map the precise lines of genetic code that are overwritten as the cell's internal logic fails. The normal cell cycle is a strictly timed sequence, divided into 4 phases of growth and replication: G1, S, G2, and M. At the transition between these phases, most notably the G1 to S and G2 to M boundaries, the cell maintains physical quality control gates known as checkpoints. Biological sensors patrol these gates, inspecting the genome for double-strand breaks or incomplete replication before allowing the cycle to advance. When damage is detected, these sensors trigger one of 2 divergent outcomes. The cell cycle pauses for enzymatic repair or initiates a terminal exit. That terminal exit is apoptosis, programmed cell death, the final method of preventing corrupted genetic data from being passed to daughter cells. If the DNA repair enzymes successfully correct the lesions, the checkpoint inhibition is lifted and the cell proceeds to a safe division. These redundant layers of verification ensure that genomic errors are intercepted before they can proliferate through the tissue. For a cell to turn cancerous, it must first hijack the body's natural growth signals. In a healthy state, growth is mediated by Proto-oncogenes, sequences that produce the proteins required for regulated cell division. A single mutational event can alter the protein structure, causing it to lock into a permanently active state. This transformation is a gain-of-function mutation. The result is an oncogene. Oncogenes force the cell into a state of continuous, unchecked proliferation, operating entirely independent of the body's external regulatory commands. The mutated protein bypasses internal logic, creating an autonomous and permanent state of pro-growth signaling. However, even with an active oncogene, the cell cannot fully transform as long as the DNA damage checkpoints are functional. These pro-growth signals are normally balanced by tumor suppressor genes, the negative regulators of the cell cycle. In 1969, Henry Harris demonstrated the existence of these regulators through somatic cell hybridization, fusing normal cells with tumor cells. Because the normal chromosomes suppressed the tumor's growth within the hybrid, the experiment confirmed that the genome contains dominant alleles that actively inhibit malignancy. The most critical of these regulators is TP53. It encodes a nuclear phosphoprotein known as the guardian of the genome. In the presence of DNA damage, TP53 halts the cycle in G1, or activates pro-apoptotic members of the BCL2 family to eliminate the cell. Cudneson's 2-hit hypothesis explains both alleles must be disabled. A mutation in the first allele leaves the cell vulnerable, but the remaining functional copy maintains control. When the second hit occurs, the cell suffers a complete loss of function. The regulatory barrier vanishes. Oncogenesis occurs at this intersection, the activation of an oncogene to drive growth, and the loss of tumor suppressors to remove the ability to stop. What forces in our environment cause these specific hits to the regulatory code? The drivers are mutagens, physical or chemical agents that alter the nucleotide sequence of the genome. Solar UV radiation causes DNA strands to physically deform by fusing adjacent bases, creating thymine dimers that lead to skin cancer. Chemical carcinogens, like those concentrated in tobacco smoke, present a persistent mutagenic threat. Compounds such as benzo(A)-pyrene form DNA adducts, bulky chemical bonds that disrupt the accuracy of DNA replication. A third pathway involves oncogenic viruses, like the human papillomavirus. These viruses introduce foreign genetic material into the host cell, often directly integrating their own DNA into the host's regulatory genes. Whether through radiation or chemical toxins or viral integration, the environment acts as the catalyst that pushes a cell past its mutational threshold. we can observe the physical results of this molecular collapse by returning to the cellular level. The cell that has lost both regulatory alleles and gained an oncogene begins to ignore all structural boundaries and apoptotic signals. Through clonal expansion, every resulting daughter cell inherits this identical, corrupted genome, creating an immortal lineage. This localized, uncontrolled division physically displaces healthy tissue, manifesting as a solid mass. The physical tumor mass is the cumulative result of thousands of localized signaling failures occurring at the microscopic molecular level. Modern diagnostics track these microscopic failures by looking for the molecular waste products of the tumor. Mutated cells shed specific proteins, known as tumor markers, directly into the bloodstream. One primary class of trackable signatures is oncofetal antigens. These proteins are normally expressed only during fetal development, but the mutated genome of a tumor often accidentally reactivates the genes responsible for them. Tracking markers like alpha-fetoprotein and carcinoembryonic antigen allows clinicians to monitor the growth and recurrence of liver and colorectal cancers. Bioinformatics uses complex sequencing algorithms to align these data streams, mapping specific biomarker levels against a diagnostic threshold. By analyzing these markers, we turn the biological chaos of a mutation into a readable data trail that guides clinical decisions. Cancer is the result of a dual-axis failure in the molecular networks that normally enforce cellular order. Mapping these specific genetic deletions allows clinicians to move from generalized chemotherapy to targeted molecular therapies. In this data, we find the specific vulnerabilities required to disrupt the disease. Subscribe to Bioinfo Bytes to decode more of the code of life.